So our second to last speaker is Anita Hopper from Ohio State, who's going to talk about parallel pathways for tRNA export from the nucleus.
Okay, so most people here know that tRNAs are small non-coding RNAs that bring amino acids to ribosomes so that translation can occur. They also have a number of other biological functions. And if tRNAs are not expressed at the right level, or there's alterations in the machinery for post-transcriptional processing, then a variety of human diseases result. So this is our working model for tRNA biosynthesis, and tRNAs, I don't think I have a pointer, okay. tRNAs are made as end-extended transcripts, and about 20% of yeast tRNAs have introns. So after the purple ends are, oh, here it is, are removed, and the tRNAs receive a number of post-transcriptional modifications. The end-matured, partially modified intron-containing tRNA leaves the nucleus to the cytoplasm, where it makes its way to the surface of mitochondria, where this tRNA splicing, heterotetrameric tRNA splicing under nuclease resides. After splicing and further modification, the tRNAs can be amino-acylated and participate in protein synthesis. And they can also constitutively return to the nucleus by a process called retrograde nuclear import. And provided that the cells are well fed and unstressed, these imported tRNAs can re-export to the cytoplasm and again participate in protein synthesis. And our genetic studies have identified a variety of proteins, three members of the beta-important family that are involved in this nucleus cytoplasmic transport of tRNAs. And LOS1 is involved in the, by genetics, the primary export, and MSN5 and LOS1 are involved in the re-export steps. But we didn't have any biochemical data that supported that, so Ivy Wang in my lab developed an in vivo pull-down experiment where we could trap nuclear export complexes and those also having tRNAs. So we made active tagged constructs and trapped export complexes by trapping RAN in the GTP form, used formaldehyde cross-linking, and then CHIP, co-IP, and then looked at the proteins that came down with these exporters as well as the RNA. Okay, so what you can see here is if we're probing for intron-containing tRNAs, then LOS1, when it's in the RAN GTP form, will co-purify with these with tRNA. Intron-containing tRNA and LOS1 co-purify, but they don't co-purify. Intron-containing tRNAs do not co-purify with MSN5. In contrast, the mature tRNAs co-purify with both MSN5 and LOS1. So this verifies the genetics, which says that LOS1 participates in the initial export of intron-containing tRNAs, and LOS1 and MSN5 participate in the re-export process. The problem is that neither of these genes are essential, and actually a combination of double mutants, our cells are very healthy, yet it's essential for tRNAs to move from the nucleus to the cytoplasm. So we took an unbiased approach to find the missing, this genetics said there must be missing exporters, and we took an unbiased approach to find those, and we used the vast resources available for yeast to probed the unessential and the essential proteome, and we extracted RNA from the 6,000 yeast strains, and then subjected each to radioactive northern analysis, non-radioactive northern analysis. This was primarily done by a single graduate student, Jingyang Wu, and a high school student, Alicia Bao, and later on with some help from some others. Okay. So we were able to look at each of the mutants and ask, is there any step in tRNA biogenesis that affected in these mutants? And we found 162 new genes that were unknown to play a role in tRNA biogenesis, and they were each verified by a variety of methods. I just want to tell you about the ones here in blue. These are the mutants that accumulate end-process intron-containing tRNA, because these are candidates for mutants that are defective in tRNA nuclear export. And among these, we found 3 essential genes, MEX67, MTR2, and CRIM1. And what's known about these is MEX67 and MTR2 are known messenger nuclear exporters, and CRIM1 is a known protein nuclear exporter, and in vertebrate cells plays a role in a stress-related mRNA nuclear export for a series of mRNAs. So these gene products that have previously been implicated in messenger and protein nuclear export seem to accumulate tRNAs in the nucleus. So here's the data showing that they rapidly affect, at the non-permissive temperature, the accumulation of intron-containing tRNAs. If we do fluorescence in situ hybridization, you can see at wild-type cells, tRNAs are distributed throughout the cells, but after a 2-hour exposure to the non-permissive temperature, mutants accumulate tRNA in the nucleus. So they're defective in tRNA nuclear export. Remarkably, MEX67 MTR2 can completely suppress the deletion of the known tRNA nuclear exporter. So when it's deleted in loss-1 cells, intron-containing tRNAs accumulate, and if we just overexpress this heterodimer about 5 to 20-fold, there's no longer accumulation, and tRNAs no longer accumulate in the nucleus. So experiments like these have led us to conclude that MEX67 and MTR10, the messenger nuclear export systems, and CRIM1, the protein nuclear export machinery, are also involved in tRNA nuclear export, shown both by the accumulation of the unspliced tRNAs, the nuclear accumulation of these tRNAs, and the genetic interactions with the known exporters, leading us to suggest that there may be 3 parallel pathways for tRNA nuclear export. Okay. So in the remaining time, I'd like to talk about another category of mutants that also accumulated unspliced tRNAs, and we're proposing that these are defective in delivering the splicing endonuclease to the surface of the mitochondria. And we're interested in this because the mitochondrial surface, cytoplasmic surface, seems to be a warehouse for RNA processing. I told you that the tRNA splicing endonuclease in yeast is housed there, but in metazoan cells, the pi-RNA processing, 5' and 3' processing machinery is also located there. So we found a couple genes that affect other genes. These are unessential genes that affect the processing of removal of introns from precursor tRNAs. As you can see here, Tom70 accumulates intron-containing tRNAs, SAM37, and they're suppressed, they're complemented by the wild-type copy. Unlike, oh boy, that doesn't show up. You'll have to take my word unless it looks better there. Maybe it does. Unlike the mutants I told you about a little bit earlier, the exporters, in these mutants, tRNAs do not accumulate in the nucleus. They accumulate in the cytoplasm as if the step of splicing that's effective is post-nuclear export. So these two proteins I'm telling you about are actually components of the outer mitochondrial surface, and they're involved in sorting of proteins to the mitochondrial interior. And this led to the model that these two proteins, Tom70 and Sam37, might be involved in assembling the splicing endonuclease heterotetramer on the outer surface of the mitochondria. And so that when these proteins are absent from yeast, then perhaps the splicing endonuclease does not efficiently assemble on the surface of mitochondria. So to test this model, we endogenously tagged the Tom70 and Sam37 proteins, and they're totally active. And then we looked at the, we tagged the splicing endonuclease subunits, all 4 of them, only 3 of them are shown here. And then we looked at where these proteins are located in wild-type cells and Tom70 and Sam37 cells. So in this, green is where the particular splicing endonuclease subunit is located, and red is DAPI staining, and the big circle here is the nucleus, and the dotted circles here are mitochondrial genomes. So you can see that in wild-type cells, splicing endonuclease subunit 15 co-localizes with mitochondria, as shown in the yellow. And likewise, so does SEN34 and SEN54. This is already known that these splicing endonuclease subunits assemble on the mitochondrial surface. And hopefully you can see here that in a cell missing this mitochondrial outer membrane protein, SEN15 does not efficiently locate to the surface, but it does in SAM37. And conversely, in first SEN34 subunit, the 34 kilodalton subunit of the splicing endonuclease, the proteins aggregate in spots in SAM37, but they do not, these spots do not co-localize with mitochondria. So they are defective in getting to the mitochondria in SAM37 mutants. So each of these gene products, Tom70 and Sam37, seem to be involved, but maybe in different ways, to assemble a splicing endonuclease heterotetramer on the surface of mitochondria. Okay. So in summary, our unbiased genome-wide screen for defects in tRNA biogenesis have uncovered 162 novel genes. 3 of them are now implicated in the delivery of precursor tRNAs from the nucleus to the cytoplasm, and 2 of them are now implicated in the assembly or the targeting of the tRNA splicing endonuclease complex to the mitochondrial surface. So I want to acknowledge the people who've done this work. Jingyang Wu and Ivy Wang were previous members of the lab who did the in vivo biochemistry of the export complexes in the genome-wide screen that I described, and Kanal, Yao, Shubra, and Alicia are currently working on the projects for the unpublished data I showed you. I want to acknowledge the NIH, my department, and actually SGD for all their wonderful resources. May they be long-lived. Thank you very much.
Time for maybe one question.
Time allows
So the Tom70 and Sam37 are unessential. The cells do not grow well in glycerol minus mutant, but they still have mitochondrial genomes. They haven't lost them.
Maybe one more
So,
quick question.
I have a question about the screen. So, how many of the hits you got are the component of nuclear pore complex?
Yeah, so previously it was known that from the HERT lab that there were a number of nucleoporins involved in tRNA nuclear export. We found 5 additional ones, and they are at every part of the nucleopore, on the fiber, the and the basket. So we hit every single part of the nucleopore complex.
Okay,
And there's some that, there are some that don't seem to play a role, and that could be due to their redundancy, as has been shown by the Wente lab.
Okay. Okay, thank you.
Thanks very much, Anita. Right.